Assalamualaikum and good day everyone. Today we are going to delve into a fascinating study on the impact of different heavy metal concentration on water levels.
Copper, an essential trace element for life, can become a destroyer when its concentration in water becomes uncontrolled. Copper mines discharge industrial wastewater into ponds, causing copper pollution. This leads to chlorophyll degradation in aquatic plants and root necrosis. The habitats of mayflies and fish are destroyed, ultimately causing the entire body of water to lose its vitality. However, water lettuce can purify copper from water. Water lettuce has a vast root system that can physically absorb copper ions. However, when the copper ion concentration reaches a certain level, excessive copper ions can cause water lettuce to lose water and wither, and roots to break and rot.
Alright, the main problem addressed in this study is the contamination of water by copper ions. These ions are commonly released from industrial wastewater, mining activities, and agricultural runoff. At high concentration, copper ions become toxic to aquatic organisms and can threaten both ecosystem and human health. Although several conventional treatment methods exist, many are costly and may create secondary waste. Therefore, our study explores the use of water lattice as a natural, low-cost, and environmentally friendly solution for removing copper ions from contaminated water through phytoremediation. Before discussing our experiment, let's first understand the problem. Copper ions are released into water through industrial wastewater, mining activities, metal processing, and agricultural runoff. While copper is essential in small amounts, as excessive levels become toxic to aquatic organisms, damaging fish, plants, and microorganisms, and disrupting the ecosystem. To address this problem, we choose water lettuce, a fast-growing floating aquatic plant with long fibrous roots that can naturally absorb heavy metals from water through phytoremediation. Water lettuce is also able to tolerate copper pollution by storing excess copper mainly in its roots and producing antioxidant enzymes that reduce metal-induced stress. These natural adaptations make it an effective, low-cost, and environmental-friendly option for removing copper ions from contaminated water.
Heavy metal pollution is one of the major environmental challenges affecting aquatic ecosystem worldwide. Among these pollutants, copper is widely released into water through industrial activities such as mining, electroplating, and wastewater discharge. Although copper is an essential micronutrient at low concentrations, excessive levels become toxic. High concentrations can inhibit plant growth, damage root systems, and reduce overall water quality. Therefore, developing a sustainable and environmentally friendly methods to remove copper from contaminated water is becoming important. One promising solution is phytoremediation, which uses plants to naturally remove contaminants from the environment. Before discussing our study, let's first understand the concepts of bioremediation and phytoremediation. So, bioremediation is a biological approach that uses living organisms, such as microorganisms, to remove pollutants from the environment. On the other hand, phytoremediation is a specific type of bioremediation that specifically uses plants to remove or stabilize contaminants. So, in this study, we selected the water lettuce plant because it grows rapidly, has an extensive vaporous root system, and is highly effective at absorbing metals from water. So, now that we have introduced water lettuce as our phytoremediation plant, let's look at how it actually removes copper from contaminated water. So, as you can see, the phytoremediation process begins when copper ions are present in contaminated water and come into contact with the root surface of water lettuce. First, the copper ions are adsorbed into the outer surface of the roots. They are then absorbed into the root cells through passive and active transport mechanisms. Once inside the plant, most of the copper is accumulated within the root tissues, which helps reduce its toxicity. Only a limited amount of copper is transported from the roots to the shoots through the xylem. As a result, the concentration of copper ions remaining in the water gradually decreases, demonstrating the phytoremediation capability of water lettuce. So, this mechanism has been reported by several previous studies. The following literature review summarizes the findings that support our research. Therefore, our objective was to investigate the effect of different copper concentrations on the growth and health of water lettuce. Specifically, we compared plant growth under different copper concentrations, observed toxicity symptoms, and evaluated changes in root and leaf biomass throughout the experiment. A study by Leo and colleagues compared 8 aquatic plant species for copper removal. They found that water lettuce showed a high copper uptake capacity, with most copper accumulating in the roots. However, increasing copper concentrations also reduced plant growth, highlighting both its phytoremediation potential and its sensitivity to copper toxicity. So, similar studies were reported by other researchers investigating water lettuce under different experimental conditions. One more study is by Butra and colleagues demonstrated that water lettuce effectively removed copper and lead from contaminated water, with most metals accumulating in the roots. More recently, Campbell and colleagues also showed that water lettuce could also remove multiple heavy metals from wastewater. However, both studies reported that higher metal concentrations reduced plant growth and overall remediation efficiency. So, building on these findings, our study focused specifically on evaluating the effect of different copper concentrations on water lettuce plants.
This is the first method we used. We prepared 5 different copper solutions running from 1 to 5 milligrams per liter with 3 replicates for each solution. We measured the absorbance of the solutions every 2 days to determine if the water lattice could clean the copper solution. We use the average values to ensure the data is reliability. Our prediction is that concentrations below 2 milligrams per liter may not affect plant growth, while concentrations above 2 milligrams per liter may cause plant dies, and the copper concentration in the solution may decrease over time. However, in practical, there was a technical difficulty. The original absorbance measurement could not be executed according to the plan. We added 2 ml of test solution, 2 ml of 5 NaOH, and mixed, then measured the absorbance at 600 nanometer. We hoped to calculate the remaining copper concentration from the absorbance change. However, when we measured, we found that the absorbance of the solution was too low, even negative. This was because our copper solution concentration was only 1 to 5 milligrams per liter, and the concentration of the complex formed was too low, exceeding the detection limit of the spectrophotometer. To address this problem, we tried to increase the copper concentration in the test solution. We add 2 ml of 300 milligrams per liter copper solution to the test tube. We hoped that increasing the total copper amount to increase the absorbance. However, the results were still unsatisfactory. Even after adding a high concentration of copper, the absorbance still too low, and the minute concentration changes in the sample could not be detected. Therefore, we still cannot calculate the remaining concentration of copper solution. We also considered using more precise instruments, such as the AAS. However, it is too expensive, and our budget doesn't allow it. Therefore, after group discussion and with the doctor's approval, we abandoned measuring absorbance. We changed our experiment objective to observing plant growth and water quality changes. This experiment can also reflect the effects of copper on plants. For the improved method, we expanded the concentration range to 1 to 20 milligrams per liter. This allows us to observe at which concentration copper begins to cause plant dies. Since no need to measurement data, repeated experiments are unnecessary. We observe and photograph the plant's condition and water quality daily, such as whether the leaves are yellowing or wilting, whether the roots are rotting or broken, and whether the solution is cloudy. After 11 days, we weighed the plants and compared the weight to the initial weight to analyze the effect of the copper solution on the plants. In this method, first we labeled the beakers and prepared 20 different concentrations of copper solution. Then we weighed the water lattices and placed them in the beakers. After 11 days, we separated the roots and the leaves of the water lattices. Then we weighted the water lattices again.
Moving on into our experimental observation, let us examine this slide, which is document the morphological appearance of our water lattice plant across the lower half of our copper concentration gradient, specifically from 0 to 10 mg per liter. It was observed over our 11 days incubation periods. At the far left, you can see our control group, which was exposed to the 0 mg per liter that was representing the pure distilled water. We can notice how the plant remained highly vibrant, exhibiting the healthy green coloration, and also have a dense fibrous root system beneath. When we move to the 4 mg per liter, the plant continues to show a remarkable structural integrity with a minor yellowing at the very tips of the older lower leaf. It was confirming that the water lattice has a robust natural tolerance to a low concentration of copper. However, as we step up to 6, 8, or 10 mg per liter, we observe a steady and visible progression of psychological distress. The leaf margin begin to lose their green pigmentation, transitioning to a pale yellow, which indicate an onset chlorosis. Underneath the water, the once fluffy roots are turning black and brown color. It was starting to look thin and fragile. This slide clearly demonstrates that why the water lettuce is resilient, but the psychological tolerance is put under pressure when the copper level rises toward the 10 mg per liter. So, if the before slide show an onset distress, this slide reveals a catastrophic physiological collapse of our plants as we go higher into the concentration range from 15 to 20 mg per liter. At this elevated copper level, the visual symptoms of toxicity are immediate, severe, and also irreversible. As you can look at the image, plants exposed to 15, 16, 17, 19, and 20 mg per liter, the healthy green coloration is almost entirely gone. Instead of the leaf, shows extreme chlorosis, with the many tissue turning into the bleached yellow or a dark necrotic brown. Necrosis represents the actual tissue damage or death, and you can see that the leaf is structurally collapsed, wilting, and decaying. But the most dramatic change is occurring beneath the surface. The root system of this plant have suffered near complete browning and decay, and a major portion of the root mass have physically detached and shed into the water. This phenomenon is a root shedding. It was a classic defense mechanism that backfired under severe mental distress, such as the plant's attempt to cut off the tissue that is absorbing the toxic metal, but ultimately lose its primary support structure. Consequently, the plant in this high concentration beaker become a highly turbid and cloudy, filled with decaying plant fiber and organic debris. This visual evidence strongly that suggests above 50 mg per liter is a lethal zone for our water lettuce. To transition our visual finding into scientific data that we can analyze, we developed a standardized scoring system shown here. In our study, we need a consistent way to quantify the daily quantitative change in the observed health leaf and also water clarity. To do so, we establish a scoring matrix ranging from a positive 3 to a negative 6, where we can see a leaf that necrosis, a total shedding, a highly turbid water. This rubric allows us to score each plant beaker daily by translating these qualitative indicators such as marginal yellowing, necrotic patches, and also water cloudiness into a specific numerical rating. We are able to calculate the mean score rating and objectively map the health of our plant over the 11-day experimental timeline. This systematic scoring key is critical because it bridges the gap between the visualization, observation, and also the vigorous statistical analysis, which is enabling us to plot the precise rate of our plant decay across 21 beakers. So let's see the graph 1.1, which plot our qualitative leaf health and also the water turbidity score across the different copper concentration of a full 11-day experimental period. This graph tells us a very clear story of time and concentration dependent toxicity. If you look at the top of the line, where the 0 mg per liter maintain a flat line across +3 throughout the entire 11 days. It was representing a perfect health. Our low concentration group, particularly at 4 mg per liter, show a gradual and minor decline. It was ending the experiment with a positive healthy zone. But if you look at the steep downward trajectory of the curve representation concentration of 10 to 20 mg per liter, within the first 2 to 3 days, their health score plummeted into the baseline of 0 and entering the negative territory, indicating an active damage. By day 11, the 15, 17, and 20 mg per liter treatment have bottomed out near absolute minimum of -6. This rapid decline in health directly correlates with the surge of water turbidity as the root decay and organic matter leaching the clouds to the water. The sharp slope of this curve highlight the high copper concentration do not just cause the damage, but they cause the rapid and irreversible tissue death within matters of days. Enough with the qualitative scoring. Now let's moving on to the quantitative measurement, where the table 2.1 show our raw gravimetrics weighing data. To understand how the copper impact the overall growth and resource allocation of the plants, we weigh each of the 21 water lettuce plants before placing them into the respective beaker. And when we harvesting them on the day 11, we are carefully separating the plant to the leaf and roots component before taking their final fresh weight. This separation is vital because the roots and the leaf of the aquatic plants play a vastly different roles in the heavy metal phytoremediation. The roots act like a primary interface, absorbing the copper into the surface and absorbing it into the cell, while the leaf is responsible for maintaining photosynthesis. By comparing the initial whole plant biomass with the final segregated biomass of the leaf and root, we can determine not only the overall growth rate, but also how the copper stress forced the plant to shed the biomass or even shifting its resource allocation. This quantitative data provide us the mathematical proof that the several growth inhibition and physical root loss that we observe visually. In this graph 2.1, it was visualizing the final difference in biomass for both the leaf and the roots across our concentration gradient. This graph provides a stark mathematical representation of copper toxicity. On the y-axis, we have the difference in weight in gram, where positive value indicate a net weight gain over 11 days and a negative value represent a net loss of biomass. Our control plant, which is at 0 mg per liter, show a strong and healthy growth. As we move to across the x-axis at a higher concentration of copper, we can see that the weight difference dropped sharply, around 15 to 70 mg per liter. The growth rate flattened because it indicates the plant have stopped growing and also struggling to survive. Once we exceed the 70 mg per liter and enter 90 and also 20 mg per liter, it was plumaged deep into the negative value. This negative value especially severe in the roots because it reflecting the physical loss of the mass due to root shedding and also decaying. This qualitative graph prove that the high copper concentration leads into the severe net loss of the plant's tissue, completely arresting the plant's ability to grow and also accumulating the biomass, which is a major implication of this viability of this plant for the long-term wastewater treatment. Okay, so let's now discuss about the underlying physiological mechanism and ecological constraint that explain this observation. The primary driver for our result is the dose-dependent toxicity of the copper. While the copper is essential micronutrients, when the concentration is exceeding the plant tolerance threshold, which is 3.59 mg per liter, it triggers a massive accumulation of reactive oxygen species, or ROS, in both root and leaf tissue. This oxygen radical initiates the lipid peroxide, which is directly attacking the cell membrane of the root. This membrane damage leads into the electrolyte leakage, tissue necrosis, and also ultimately that causes the root to darken, weaken, and also physically detach from the root plant, a phenomenon that we saw during our observation. So in the leaf, copper translocate from the roots accumulate into the chloroplast, which is disrupting their internal structure and also degrading the chlorophyll alpha and beta pigment. These inhibit the photosynthesis electron transport, therefore starving the plant of carbohydrate and producing the severe leaf chlorosis and necrosis as we documented. Finally, the dramatic rise in the water turbidity is driven by the organic leaching. As the plants die, the cell and the root decay, they release organic compound and protein into the water, which stimulate the bacterial growth and also clog the beaker. This is the highlight of the critical phytoremediation constraint. If the copper concentration exceeding the tolerant limit, the plants will decay, the releasing metal back into the environment and then causing the secondary pollution.
Alright, copper can be dangerous to the environment because excess copper ions are toxic to aquatic organisms, plants, and microorganisms. They can reduce water quality and disrupt the balance of ecosystem. To prevent environmental contamination, copper-containing waste should never be poured down the drain. Instead, it should be collected separately and disposed as a hazardous chemical waste according to proper safety regulation.
So, to conclude, as shown in the slide, we have successfully achieved our objective by systematically evaluating the morphological and also biomass-related effect with the different copper concentration on the water lettuce. Our finding established that the water lettuce is a highly effective, low-cost, and also sustainable phytoremediation agent under a low-cost under a low concentration of copper regime, specifically under 5 mg per liter. However, it suffers severe psychological distress, tissue necrosis, and root shedding when it was exposed to higher concentration of 10 to 20 mg per liter, making it a double-edged sword. While it was served as excellent biological indicator to the metal pollution due to its rapid and visible morphological changes, its practical deployment in industry wastewater treatment is limited by the strict toxicity and the plant will decay, also compromise the water quality, increasing the turbidity, and also releasing back the metal into the environment. Not to forget, this is the reference list that we use throughout our mini project. And finally, I would like to thank you all for your attention, and for those who are involved in our mini project, whether it was directly or indirectly. Thank you so much.